Hello, Miss. So in this video I'm going to explain my knowledge about meiosis, what after reading and investigating different pages. So first I'm going to start with a definition of meiosis by my point of view. So I can say that meiosis is a relation with mitosis, but the difference is that in meiosis, meiosis is used for a purpose in the human body, just one, that is the production of gametes, sex cells, or sperms and eggs. The goal is to make the daughter cell with exactly half as the chromosomes as the cell that starts the meiosis. Meiosis in human is a division process that take a diploid to a haploid. So in this video I'm going to explain the phases of meiosis, the phase 1 and the phase 2, and how it can be related with mitosis. So let's start. So as I said before, in meiosis, meiosis is a lot like mitosis, and they make a relation. The cell goes through similar stages and uses similar strategies to organize and separate the chromosomes. A very important thing that we have to know about meiosis is that there are two stages, meiosis 1 and meiosis 2. So the first stage is meiosis 1. So I'm going to start with the first phase that in this case is called the prophase. This is prophase 1. So to start the explanation, this is a diploid. So we start the meiosis with a diploid cell. This is a homologous. They are shown here as pink and purple to differentiate each chromosome. So in this phase, the homologous chromosome pair up and exchange the fragments. So these two pair up and exchange each other by crossing over each of the chromosomes. So the pink cross with the purple, and yeah, that's basically it. So then we have the metaphase 1. This is the metaphase 1. The metaphase, we can see the homologous different. These homologous pairs and line up in the metaphase plate. When the homologous pairs line up at the metaphase plate, the orientation of each pair is random. So then we have the anaphase. This is the anaphase. This is the third phase, and the homologous separate opposite ends of the cell. Like we can see, this separates this and are the opposite each other. The sister chromatids stay together. What are the sister chromatids? This pink and pink, purple and purple, to show the sister chromatids in a simple way. Then we have the last phase of meiosis 1, that is the telophase 1. So here we have the telophase 1. We can see that is very different of the other phases. So you remember that at the beginning of the video I said that the objective of the meiosis is changing the diploid cell to a haploid cell. So this is the diploid cell. We have two phases to change to a haploid cell. So this, the telophase, is a haploid cell where each chromosome has two sister chromatids. We can see two sister chromatids of the purple color, two sister chromatids of the pink color, in a simple way, but are non-identical. So this basically is the meiosis 1, and then we are going to have the meiosis 2. Okay, so in here we have the meiosis 1, and we are going to explain the meiosis 2. So at first we have the prophase 2, that in this case will be these two cells. So you remember that in meiosis 1 the first cell was a diploid cell. So here the star cells is a haploid cell made in the meiosis 1, made in this stage. So this is a haploid cell, and in this phase what it happens is that these chromosomes condense. We can see that here the colors are separated, pink and purple are separated, and here they condense. We can see that the pink have a little of purple and the purple have a little of pink. So yeah, then we have the metaphase 2. Here we have the metaphase 2, and in this phase the chromosomes line up at the metaphase plate. So yeah, they're lined up and are condensed at the same time. Then we have the anaphase 2. Anaphase 2 here. And in this stage, the sister chromatids that we see in the phase 1 separate the opposite ends of each cell. They are separated in this phase. And then we have the last. Okay, so here we have the tail phase. We can see that we have the tail phase 1 and the tail phase 2. In the tail phase 2, there are formed new gametes as a haploid, and each chromosome has just one chromatid. So, okay, you can see there that in the first telophase there were 2 chromatids of each color, 2 chromatids of pink and 2 chromatids of purple. And in this telophase 2 there is just 1 chromatid of purple and 1 chromatid of pink. So yeah. Okay, so in the telophase, nuclear membranes form around each set of chromosomes, and the chromosomes decondense. Cytokinesis splits the chromosome sets into new cells, forming the final product that in this case in meiosis are 4 haploid cells in which each chromosome has just 1 chromatid, as we can see in the drawing or in the diagram. Okay, so now we know that this is meiosis 1, where the diploid changed to a haploid, and this is meiosis 2, where the cells are haploids, and the telophase form the gametes. But all this is called meiosis, and is the process that led the meiosis happened. So I'm going to explain in a brief way that how meiosis mixes and matches each gene. Okay, so as we know, the gametes produced in the meiosis are all haploids, but they are not genetically identical. They are different. There are many potential gametes types than just the 4 shown in the diagram, even for a cell with only 4 chromosomes. There are 2 steps. The crossing over, that is where the points where homologous cross and exchange the genetic material, and then they will be different through the meiosis. If this meiosis happens many times, crossovers will happen at many different points in the meiosis. And then there is the random orientation of the homologous pairs, that is when the homologous pairs in the metaphase 1 allow the production of the gametes with many different assortments of the homologous chromosomes in the phase 2, forming all the meiosis. Mitosis, there are produced two diploid somatic cells that are genetically identical to each other and the original parent, but in meiosis there are produced four haploid gametes that are genetically unique from each other, so they are different completely, and the original parent. Mitosis involves one cell division, where meiosis involves two cell division.